So today I'm just going to show you how we use prepared microscopy slides across the whole of GCSE biology. A lot of lab suppliers sell these complete sets of microscopy slides, which is great if you're a hobbyist or you're looking at one-to-one teaching, but having one of every slide is completely useless for a classroom setting. This is on Amazon, but a lot of our lab suppliers do sell very similar sets to these. And we used to actually give out these sets when teachers would ask for introduction to microscopy or when they're covering specialised cells. But when you actually look into what's in them, half of them are pretty useless and can't be used for anything that we cover in our course. And the ones that we could use, there's only one of. So it's hard to get that round the classroom. And then point out to the students the structures and features of that tissue that they should be looking at. Since the new specification abolished coursework, students are now expected to be able to remember the parts of a microscope, know exactly what they do and how they're used. And so the more opportunities we can give them to get a microscope in their hands is only going to help them pass their exams. So here's some of the changes we've made in our department to how we do pre-prepared microscope slides. So after doing a short course in histology, pathology and microscopy, I soon realised what we actually needed was sets of 15 or 20 of the same slide to be able to give to every student and then be able to walk the whole class through that slide and almost give them a tour of that slide, pointing out all the features in structure and function of those cells and those tissues. So this is a really great one to start out with. This is just human blood smear. So just contact your lab supplier, contact your rep and tell them that you're looking for 20 of the same slide. Ideally, you want exactly the same slide from the same batch, all stained in the same way. Something like blood is going to be really easy and universal, but it does get a bit more complicated when you get to things like more complicated tissues and organs. You might even be able to find half of a set already. So this set, I actually only ended up buying 10 because I already had about 8. So this is how a blood slide is prepared. You take a drop of blood, you take another microscope slide and you actually use it to scrape it across the first slide. And you can actually do this with the students as a practical, but they have to be willing to lance themselves and they have to work with their own blood. But if they've made them or you've ordered them, you should end up with something like this, where you've got a very dense end just after the application point, and you've got a very less dense end where the cells are too spread out. And then that sweet spot is near the end where the cells are nice and spread out, but not too spread apart. So this is what one might look like. This is using an online photo stitching program, which allows you to stitch multiple images together so the students can see lots of detail and can be shown to the students really zoomed in, looking at individual cells or zoomed out at a macro view. So looking at this from a macro view, you can kind of see the left side that's pretty dense and the right side's a little bit lighter. So we want to be looking sort of around there. That's where the even distribution of cells are going to be. So we're going to zoom in and we can start seeing some cell differentiation there. So we can start talking about stains. So this is right stain. It stains nuclei, dark purple, but it will stain cytoplasm a light pink. This is really great for when we're talking about the lymphatic system, we're talking about the immune system. We can see some platelets there. We can also see some white blood cells. We can differentiate those white blood cells. We can see some lymphocytes, some macrophages, some basophils, some eosinophils. And we can talk about how we count those white blood cells and do a white blood cell count. And zooming in, we can start seeing that classic donut-shaped red blood cell. We can start talking about their surface area, and we can talk about how all of these cells are specialised for their job, so really lovely for teaching specialised cells. Lots of students actually enjoy looking at blood and organ tissue. It gives it that kind of gross factor that a lot of students actually enjoy from biology. And this is actually an exam question taken straight out of a separate science paper. So here you can see those white blood cells, the red blood cells, and it literally is the same diagram as what they would see on a blood smear. And these blood smear slides are really cheap. They're only maybe £6 or £7 each. So to get a set of them, especially if you've already got some, can be quite economical compared to other slides. Currently, lab suppliers only sell them as singles, so you have to buy them in sets of singles. But if any of the lab suppliers are listening, hopefully you cotton on quick and start selling these sets. I feel like you're missing a massive trick here. So over time, I've accrued these sets and built up these sets. I've got sets for blood, lung tissue, nervous system, specialised cells, botany, testes, heart tissues, root hair cells. Can be a really nice short 10-15 minute activity to break up a single lesson, or even a longer activity if you wanted to add some drawings and estimate cell sizes. Okay, so let's look at some other tissue. This is lung tissue now. So this is a really nice example of sectioning. So this has been sectioned transversely, horizontally across the lung. And we can draw attention to the fact that specimens need to be cut ultra thin, 1 cell thick. Zooming in straight away, we can see those bronchioles. So I really like showing this with lung dissection demonstration, where they actually feel the different textures of tissues in the lung tissue and the trachea. And we can actually see that difference of texture in the bronchioles and the lung tissue itself. So here we can see some light, fine lung tissue in comparison to something like these cartilage rings that are much darker and denser, and that relates to structure and function. Now this part's my favourite part, so zooming in, we can actually see individual alveolus. Massively different to what a textbook would look like. This is stained with H&E, which is a pretty universal stain, so light pink cytoplasm, dark pink purple nucleus, but it stains blood a kind of red orangey colour. So if we look really carefully, we can see that network of 1 cell thick alveoli, and we can actually see how delicate this tissue is. Really great for teaching specialised cells, tissues, organs, organism, and you can use it in conjunction with blood and the heart when teaching circulatory system. And in each ring, you should be able to see a red or orange dot, which are going to be those capillaries that facilitate the diffusion of oxygen and carbon dioxide between the alveoli. So these are some textbook diagrams of lung tissue. You can have the textbook open next to them, you can have them printed out, or you can have them on the PowerPoint on the whiteboard. And also illustrates really nicely that what you see in a textbook isn't necessarily exactly what you're going to see in real life. But it's a really lovely opportunity for students to see real lung tissue and those specialised cells inside the lung. All right, now we're going to look at the kidneys. The kidneys are a really great slide for triple science or for A-level, and again goes really nicely with the kidney dissection. So we've got a longitudinal section here, cut long ways, and we can already see that classic kidney bean shape. We can start seeing some changes in texture here, indicating that there's a change in structure, which therefore means there's a change in function. So let's have a look now at low power. We can really see that change in texture there. So renal cortex around the outside, medulla in the middle, and then go into the ureter as you get to the center. Lovely opportunity for them to see Bowman's capsules. Does really look like the textbook, so we can see these round structures of the glomerulus here. You've got those capillaries that are getting smaller and smaller and tighter in the Bowman's capsule. Now as we go down the nephron through the tubules, they've got to remember that there's millions of these nephrons, and some of them are going to be cut more longitudinal and some of them are going to be cut more transverse, depending on what they're looking at and which direction they're facing. And there's those collecting ducts going on side those loops. So we should start seeing some loops here. We won't really be able to see the loop of Henle as it is in the textbook, but we will be able to see kind of cross sections of those tubules. And then as we go further down, tubules turn into collecting ducts. We'll start seeing them get a bit more longer and wider as they get bigger, and that will be back out into the collecting duct and then the ureter. Now this is a very simplified version of what's going on here for GCSE. You can actually see the ureter right there. That's been sectioned really nicely and really well preserved. And if we're lucky, we might be able to see some blood. So we're just looking for that bright orange, red kind of patches. So there will be the bigger branches that turn into the fine capillaries, which surround the loops for reabsorption. There is loads more to look at here, but this is just a really nice example of a 5-minute activity to break up something that's really heavy in content, like the kidneys. All right, we've got something real out there now. This is rat testes. I'm not really 100% sure how this has been sectioned, but obviously the organ is spherical, and so this has been kind of taken as a quarter of it. Nice for when we're teaching reproduction, obviously a topic where there's not a lot of lab opportunities. Always getting teachers asking, have you got anything for reproduction? Nice to be able to say, yes, I've got some lovely little testes slides for you guys. But also nice further up the school if we're studying gametes or meiosis, or even in specialised cells straight after the introduction to cells topic. So straight away we can see this repeating pattern of circles. So these are the seminiferous tubules. They're going to be a kind of concertina shape of these tightly coiled tube structures, folded back and forth in themselves for maximum surface area, which is where your sperm cells are going to be produced. Okay, let's get a closer look now. So now, inside and around each seminiferous tubule, got this regular rich blood supply. You've got this barrier in the tube that's formed by Sertoli cells, which is going to give it structure and support. And in between the tubules, you can see this dark purple, which is actually the Leydig cells, which produces the testosterone. Okay, down to the nuts and bolts of it. Sperm cells are actually really difficult to see because of their size. They are really small. Smallest cell in the human body. Realistically, you need something like oil immersion, and to be honest, if you just get testes, they're a lot easier to see and you get more out of it. So what's nice about these slides is that all the sperm cells face the same way. Their heads face the outside of the tubule and all their tails are pointing inwards. Where they're so bunched together, you can see them a lot clearer and you can get a nice comparative size compared to the other cells in the area. And you can get a really nice appreciation for exactly how many sperm cells are produced and how tiny they are. Genuinely microscopic. Now we're on to one of my favorite ones. This is an entire human foot. Obviously, I don't have 15 slides of these. This is just straight from the Easy Zoom website, and I'll put this on the big displays or the big TVs that we've got in our labs, and I'll literally just walk the students through this one from the front of the classroom. And it's just absolutely amazing for teaching introduction to structure and function of body systems, cells, tissues, organs, organisms, and even specialized cells. And just what an amazing bit of science. A whole foot that's been sectioned longitudinally, super thin, one cell thick, using probably Mason's trichome stain, which I'm not 100% sure. If someone wants to correct me in the comments, please do. But that'll be red for keratin and muscle, and then blue or green for bone, light red or pink cytoplasm, and then dark brown to black nuclei. So really nice cell differentiation there as well. So let's get into this one. You can see the thick layer of the epidermal cells at the bottom of the sole of the foot. You could contrast that with the top side, which have a much thinner layer. Remember, evolutionarily speaking, we've never really had shoes apart from pretty recently. So now we can see some blood vessels, and we can also see some fatty tissue that's going to also give some added protection as well as protect the internals. And remember, collagen is stained blue, so that's going to hold a lot of it together. Then we can start seeing muscle tissue here, so that classic line layered muscle tissue. It's going to give you all the dexterity and the movement in your foot. And now zooming out, so we're just going to go across now to the bone section, so we can start seeing our bone really dense and compact compared to that muscle fiber. And then we can start seeing our osteocytes and osteoblasts, which are involved in maintaining that bone tissue. What an amazing slide. You can buy whole organ slides as well. Sometimes you can get whole eyes, whole hearts, whole lung tissue, usually sectioned from a small mammal like a mouse or a rat. But just contact your lab supplier and see what they've got. Can't talk about microscope slides without talking about microscopes. So these are the ones that we use in school. A-level biology has a much nicer, fancier set, but these are great for 7s all the way up to 11. Did cost a lot to replace all of our sets, but it was totally worth it. They've got LEDs on the bottom, so it makes it really easy for these 5-10 minute break-up activities. And the more practice they get with the microscope, the better. They've got to be able to recall all of those parts. Does mean we have to maintenance them a little bit more, but actually I don't mind doing that when I know they're being used really well and thoughtfully, and the experience they get is really purposeful. And here we have the slightly older style microscope with the mirror. Do actually keep one of these in every lab, so any teacher anytime can do a quick little recall on the parts of a microscope. Now, here's what I really want to show you. This is what I'm working on at the minute. Got this lovely package from Blades Biological. They distribute stuff in the UK for Carolina Biological, who are based in the US. These weren't cheap. They were really expensive, but Blades did do me a discount, so thank you very much. So we've got a blood smear here, and hopefully with what they already know about staining and about the composition of blood and the composition of the cells, they might be able to diagnose what is wrong with this patient. So we know that right stain will stain nuclei dark purple, and we can see that in our white blood cells, and obviously our red blood cells are unnucleated, and we can see a few platelets that have picked up the stain as well. And hopefully after a little zooming in and searching around, they should be able to notice that some of their red blood cells are showing signs of having a nucleus. And this patient has actually been infected with the malaria parasite. So we teach about malaria, we teach about blood diseases, we also teach about parasites. So this is a lovely way to marry up all of that knowledge and give them an insight into some of the work that goes on in a histo lab. If you're interested and you want to know more, you can actually find my sheet on TES. I'll link that in the description. It is a paid for one, but it took me an age to write, and it is one that we use across the whole of our curriculum. So it's really, really well used and well practiced. If you do want to have a go, I definitely recommend starting out with something like blood, or lung tissue is a really good one to start out with. I've also been messing around with frog's blood recently, but that will be a story for another time. You can write your own documents and your own sheets. It literally just takes a lot of time to research and filter out the things that we need to know in our curriculum compared to what someone would need to know if they were studying medicine or biomedical sciences. Meantime, this pack is ready to go, so we're just going to test it out in some classes and I'll keep you posted on how it goes.